So I think the crowd is small enough so that it would be worth to do a 30-second presentation, maybe like a 10-second presentation each. Just, I don't know many of the people here. I know some of them, I would say approximately 50%. So I would like maybe to call each of you, and each of you can say who you are, what your research interests are, and which institution you're working with. Okay, the next one in my screen is Pei Ling Chen. Could you please introduce yourself?
Hello, I'm a dermatopathologist at Moffitt Cancer Center. I have personal interest in cutaneous lymphoma and also other skin cancers. We have a lot of investigators here at Moffitt doing single cell research, and we have interest in spatial transcriptomics. So that's why I'm attending this session.
That's fantastic. Next one is Gary Bader. I'm sorry if I'm pronouncing it incorrectly.
Hi, yeah, that's good. I'm a computational biologist at the University of Toronto, and we're actually working on 3D electron microscopy data, but we're interested in connecting it with spatial transcriptomics data.
Yeah, that's super cool. Then we have Junil.
Hi, I'm from the University of Michigan, Ann Arbor. I'm in the medical school. I myself is working on computational methods. We also have a university-wide single-cell spatial program hoping to develop into it.
Next one in line is Alex Ropelewski.
Hi, I'm the Director of Biomedical Applications at the Pittsburgh Supercomputing Center. I'm the PI on the Brain Image Library, which is a data archive funded through the Brain Initiative, and I'm also involved in the HubMap project.
Yeah, that's cool. Then we have Cody Heiser. Cody, do you want to introduce yourself? I think Cody disconnected maybe. It's okay, so the next one is Dana. Dana, appear.
Hi, I'm from the Sloan Kettering Institute, and I'm largely a computational biologist, active in both the HCA and in the H10, and we have all sorts of imaging modalities set up, including some, the most interesting of them all. We have 3D HDR embryo imaging of up to 5 colors, which is eye-popping. And yeah, it's, we've been playing with the computation of this data type, and it's truly challenging.
Daniel Gilchrist, or Gilchrist.
Hi, I'm Dan Gilchrist. I'm a program officer at NIH at the National Human Genome Research Institute. I work in functional genomics and computational genomics on the ENCODE project, where we have some single cell attack and single cell RNA-seq data, as well as on the HubMap project.
I'll keep moving with the list. I would actually ask you to make it very short, just because I see new people are so I'm just don't want to burn all this time. Hiroyuki Aburatani.
Oh, hi. This is Hiroyuki Aburatani from University of Tokyo. We are working on single cell genomics on the cardiomyocyte and some also cancer organoid, and doing a transcriptome and attack-seq or something.
Jamie Marshall.
Hi, I'm working on spatial transcriptomics in the kidney and developing the kidney cell atlas.
Jeffrey Moffitt.
Hello, everybody. I'm an assistant professor at Harvard Medical School in Boston Children's. I was one of the co-developers of a spatial method, Merfish, while I was postdoc in Xiaowei Shuang's lab. And I use that technology pretty heavily in my own lab.
Junil. Junil Kim.
Hi, I'm Junil Kim. I'm a computational biologist from University of Copenhagen. I'm developing some tool for single cell analysis, single cell RNA sequence analysis. But recently I'm interested in cell-to-cell interaction, and that's why I'm wanting to learn some new technology in spatial transcriptomics. Thank you.
Catherine Vernon.
Hi there, I'm a postdoc at the Broad Institute, an nephrologist back in the UK, and I'm also working on the Human Kidney Cell Atlas effort.
Okay, so my list has been regenerated as Catherine was speaking, so now I cannot see. I will go with Norbert Tavares.
Hello, I'm a program manager with the Chan Zuckerberg Initiative for the Human Cell Atlas.
Ken Lau.
Yeah, hi. I'm at Vanderbilt University. We work on the H10 as well as the gut cell atlas. And we're interested in looking at spatial assays, analyzing both the microbiome and host cells.
Yeah, Maigan Brusko.
I am Megan from the University of Florida. I'm part of HubMap. We map the spleen, thymus, and lymph nodes, and we've been doing single-cell RNA-seq, and we'll be moving into spatial transcriptomics.
We have then Xin Lin.
Hi, I'm with HubMap. Our tissue mapping center is looking at the circulatory system with SeqFish.
Steve Sanson.
Hi, I'm from the University of Oxford, where my group is using single-cell methods to study inflammatory arthritis and also to make a map of human joints as part of the Human Cell Atlas.
Velina Kozareva.
Hi, I'm a computational associate at the Broad Institute in Evan McCosco's lab, and I've been working on basically the BACCN mouse brain project.
We have Vik Devgan.
Hi, this is Vik Devgan. I'm from NanoString. I'm a business lead for spatial transcriptomics.
Yes, and Xiaowei Zhang.
I think Jeff introduced me. I could save you a few minutes if I'm not introducing myself. All right.
You really need no introduction in this community.
I mean, I was going through the so my list was regenerating as people were dropping in. So if anybody wants to introduce yourself, because I just skip you, please do it now. Okay, so I can give a very brief overview, and then I really hope a lot of people will chime in. So we have a lot of technologies, including like Murphy's, Slice-seq, Visium, Transcriptomics, and so on, to measure the transcriptome. Sometimes at the level of single cell, sometimes at spots that contain 5, 10 cells, a large number of cells. So there are a certain questions that I think we should try to talk about in this workshop. So one of these questions is goals. Goals, 2 ground goals are to chart the cell type, like to spatially localize the cell type and comparison between sick and healthy. I would like people to elaborate on this topic and to maybe add a 3rd ground goal that I'm missing. A second thing would be to try to understand what are the methods that are out there that allows us to classify and to call the cell types the data. For instance, if I have a Murphy's slide, have a Murphy's spot that's at the level of single cell, and I measure 254 genes, what can I say of that cell using those genes? What methods are out there? Would you say that this is a solved problem? Would you say this is an interesting problem? And then the question moves applies to methods like Slice-seq that are not at the level of single cell, but you can still classify the single spots. And then one other question is, how do you integrate this data? And do you think it is interesting, and do you think it is a solved problem to have, for instance, one slice of tissue measured with Murphy's, one slice of tissue measured with Slice-seq, and how do we merge these 2 data set? Or even better, if you collect single nuclear or single cell, so a non-spatial data from tissue, and then can you combine this data set with Murphy's data, the data, so that you will get a data set with the same throughput of single cell and single nuclear RNA-seq data, but with special information in it. And then one other thing that I would like is people to chime in about what kind of tissue, what kind of patterns are particularly interested, and like what kind of genes and what kind of question, what kind of gene programs for like each organ, assuming we have experts working on this. And if we have time, the final thing is to review and talk about software for visualizing the data. Okay, so feel free to like change these subtopics if you want to see this workshop in a like if I forget something and I didn't include something. So the first thing I like to talk is like goals. So we are doing spatial transcriptomics because we would like to chart the cell type in the human body, and because we would like to have a information to make meaningful comparison between sick versus healthy. Would you like to elaborate on that, or would you like to add one more goal to it? Okay, the moderator will now ask some PI to comment, like Xiaowei, would you like to say something about what are your goal of doing spatial transcriptomics?
Oh, Tommaso, sorry, I cannot be at this meeting for long, so so I was just trying to be an observer here.
Okay, good.
I even have my daughter in my room. All
Dana,
right.
would you like to share some vision of?
So I have two big goals with spatial data. One is I'm interested in development, so I really want to look at morphogenesis and how tissues develop. That's where you take, sequential, and you actually see, how the tissues move with time and reorganize. My other big question is I'm very interested in how cell-cell interactions influence each other. So basically how processes are coordinated through crosstalk across cell types. So we're looking at one cell, but we don't know its neighboring cells and the signaling it receives. So one of them is to understand the cohort of cells and how they interact and influence one another. So it's really about knowing what cell is next to which and how being next to certain cells influences their phenotype, and how this sort of spatially sort of emerges into tissue level properties, which connects to the developmental story, but the first one also has sort of a morphogenesis temporal story to it.
So this is great. Like adding time and also using spatial patterns to study cell interaction are certainly topics that we should dive into more in part 2. Is there any other PI? I see Vivi here who wants to comment on the overarching goals of using spatial.
Why only PIs? The youngest people have the best ideas. They're young and innovative and fresh.
That's absolutely right. But they also need to volunteer. So would somebody like to?
I can just chime in. I agree with everything that Dana said, and that's things that we're thinking about in my lab. Can we use the local neighborhoods effectively to perturb cells and look at how they respond? So they're very natural perturbations. It's, I think, how we tend to think about the same problem she was posting. But in a lot of the tissues that we're looking at as well, there are strong spatial gradients that really give rise within a cell type to defined gene expression changes. And we're very interested in understanding how that fine-tunes the function of those cells.
So what kind of tissue do you all think is the best tissue for addressing like cell interaction, or like the goals that Dana was saying?
So I can't say one type of tissue, but I've worked with different types of tissue, and cancer is hard because it's pretty messy, And so I actually think that once you have tissue that does have clear spatial patterns, such as, for example, the colon, which has a lot of canonical patterns to it, I think starting with something that's very structured and developing methods on that is a good way to go.
So structure like as in the mouse brain, or like
mouse brain, colon. I mean, the problem with the mouse brain for the cell-cell interactions is that many of the cell-cell interactions in the brain are very long range, you know, due to all these exonics. So maybe if you want to look at the interaction one, I wouldn't necessarily go to brain, but I would go to something like colon.
We've been looking at this is Gary, we've been looking at the developing mouse brain, which doesn't have those many problems that Dana mentioned with the large neurons. And there, what's valuable is that there's experimental techniques to help test the interactions that are identified or predicted, cell culture systems and organoids.
I actually think cancer tissue is actually great for spatial transcriptomics, speaking from a cancer center perspective, because it's a great venue for studying tumor and immune microenvironment, the interaction between tumor cells and also tumor and immune cells.
I mean, I want to get to eventually cancer. It's just we've looked at a lot of cancer data, and any computational method that I can think of on the face of the planet makes very strong assumptions. And recently, I've discovered that some of my very strong assumptions are broken by some other data that I'm looking at. So I think that right now in our analysis of tumor data, and most of the published paper out there are making assumptions that are just a little bit too strong for my taste. One example is just how far the interactions can go, and just how far one cell can influence beyond its immediate neighbors. And again, lacking these canonical patterns to sort of really test your algorithms in a way that gives you confidence. I mean, you can make assumptions, you can build an algorithm, you can publish its results, and given how sexy the field is, you'll even get it published. The question is, will it be right?
So this is interesting. So we have like structured tissue and unstructured tissue, and like both of them, people have proposed both of them as like a useful like tissue that we can investigate. So one question that I wanted to ask the people here is, so once we have in situ data, we can do cell type calling on the in situ. So this means for Merfish, try to classify spots which are single cell using a few hundred genes. And for Visium, for SLISEq and spatial transcriptomics, that means try to assign a cell type identity to one spatial spot that contains sometimes like 5 to like 30 cells. So would you say that we do have methods for doing that? Do you think this is a solved problem? And if not, what are the current challenges for solving this problem?
I can start. I mean, I don't think it's a solved problem. We actually worked on this a bit, in particular in context of Merfish data. I think the underlying issue with these annotations is where you get the information. So one approach that's practical now is basically have a manually curated set of markers that are being used for annotations. That only basically works in a hierarchical manner, where you can, let's say, separate neurons from glia and so forth, and then different subtypes of neurons. And it took some tweaking in terms of algorithms. We had to develop something slightly different in terms of methods, but it can be applied to spatial data with just even like 30 markers or a few hundred markers. But it's very targeted. In essence, it depends on these pre-existing annotations that you have to put together, either from single-cell RNA-seq data or some other source. So that can be done, but beyond that sort of magic automation, we're not there yet.
So Indeed, like one issue is like to have a curated like markers for tissues like this, which we use to do this classification, and we can learn them from corresponding single cell or single nuclear RNA-seq data. But there are two other challenges, and one is the fact that some technologies do not allow for measuring too many genes, and so we might not be able to classify at cell cluster level or at very fine level just because the throughput is not good enough. And some other times we have throughput, but we don't have a resolution at the level of single cell. So one more specific question than the one I previously asked is, how do you think, do you think it's a solved problem? Or if not, what is the state of deconvolving single cells from in situ data? So for instance, I am picturing, we take slide-seq or we take Visium, and we have one, and we know there are 5 cells, and we know the expression level of that spot. Can we deconvolve that spot into, the transcriptome of the five cells?
Again, this is just something we've been playing with. I mean, you can do it to some extent, but it depends on how well you understand the composition of the tissue. And then it's also a question of what you get from this. So you can use even basic methods like ICA to do the convolution, let's say, of slide-seq. Works reasonably well. I mean, there's still some cells that are uncertain, but then it's a question of how much you believe that and what do you use the convolution for. So if you look at the original paper, they basically didn't want to deal with this kind of fuzzy data, and they just threw out those beads, right? And they could afford it because they had quite a bit of it. So I think my sort of return question is, what do you need that deconvolution for?
So I might try to answer this question, but I invite other people to answer that too. So you might have a very granular or rare cell type, and so you might not be able to find that cell type if you are not at the level of single cell, because it gets hidden in a spot with more popular cell type. So if I'm able to deconvolve, or if I can use as reference like corresponding single cell data, I might the statistical distribution of that rare granular cell. But maybe somebody else also wants to answer. And I think also Maigan Brusko asked a question in chat, and if she wants maybe to speak and ask the question to everybody, that would be cool.
Yeah. So the size of a spot and the number of cells in that spot, depending on 5 cells in tissue, might be very similar to each other, and you may have 5 of the same cells in that size spot. But in other tissues, you would have potentially 5 different cells within the same spot, depending on what area of the tissue you are working with. And so I work with the spleen, thymus, and lymph nodes. So those are fairly heterogeneous tissues with kind of defined functional areas, germinal centers, lymphoid follicles, things like that. And when we've talked about moving into this, we need to have really strong benchmarks on cell types using our single cell data before we even move into spatial data, because we have to be sure that we can define, cells that might be functionally very similar, different types of T cells that are developing, but that may be also functionally distinct at certain levels. So we just kind of are having to integrate multiple data types, I think, and I think that's what we need to keep doing, is we can't just depend on one data type to do cell type annotation or anything like that.
That was awesome. Thank you. And does anybody else want to answer or contribute to Peter's question?
Well, I can just clarify maybe what I meant, because I think if you asking for an algorithm that will have uncertainty in the answer, right? Because we can't say for sure which cell types are there. Then I think it's always useful to think through what you're going to use this for. So for instance, if you're just trying to predict likely density of the cell spatial distribution, that's one answer, and we can design algorithms for this. However, if you're trying to, let's say, infer that 2 cell types tend to co-occur, That's kind of a different analysis, and you can still do it using these types of deconvolution tools, but you basically need to start looking at these kind of probabilities directly. So it's, I think it's of interest to methods developers, you know, what types of downstream questions will be asked from this result.
So for the scribe, I think one topic that we totally need to talk about in part 2 is the co-occurrence of cell types and how that that can contribute the overarching goals of spatial transcriptomics. So one other question is, yes, please.
I think that was just noise.
Okay, so one other question is whether we can use non-spatial technologies like single-cell, single-nuclear RNA-seq to surpass, to win the limitation of spatial data. So either essentially to map single-cell, single-nuclear RNA-seq data back to space so that we can increase throughput of technologies like MERFISH, or like help or assist a decom process, and so push Visium or SLISEq at single cell resolution. Do you think this problem is solved? If not, what do you think are the technical challenges, and like what is of the art?
I'm happy to toss a few comments in here. I mean, I think it's a bit of a straw man to say it's solved. I think that these are not solved problems at all, but there's some people that have, or some approaches that have been put in place, and plenty of room for continued innovation. I think that the first thing I would say is that a lot of the image-based methods are now pushing up the multiplexing to very high levels. So there are trade-offs when you think about pushing to many genes in terms of the throughput, but you have the capacity now to make measurements at almost whole transcriptome scale, and perhaps very soon transcriptome scale. And then I would say that there are a couple ways in which the untargeted dissociated cell techniques like single cell or single nucleus sequencing can actually be very informative. They can be used to help design panels, and there have been several ways that people have developed to do this, but I think there's a lot of room for innovation on ways to extract the most informative genes from single cell data. I think there are also disadvantages there that reflect the fact that a lot of the some of the image-based methods are actually far more sensitive than the dissociated cell methods. And so there are genes that don't show up as informative in things like single cell RNA-seq, but actually are quite informative when you have the capacity to measure them with a very high detection efficiency. So those methods where you design panels leveraging untargeted methods, I think it's important to keep in mind some of the technical differences between these approaches. And then I think there's some very interesting ideas and opportunities to do joint analyses of these data. If you have an image-based single cell data set like MERFISH or in-situ sequencing, and you have single cell data, instead of using one to just inform the other, are there ways to develop analysis approaches that jointly analyze those and allow propagation of information in both directions between the data sets? I know that there are many groups working on this. I think it's a very exciting avenue for future development.
Yeah, so this is Shanwick, and I would just add a little bit to what Jeff said. So he's absolutely right in the sense that the number of genes that can be measured by imaging-based approaches is actually not that limited. And if you're interested in many thousands of genes to be measured, we could do that. But the trade-off is, of course, so when you're talking about a very large cell atlas of a large organ, then, you know, when you need to measure more genes, it's slower. You know, you still want it to be having a high throughput so that a large volume can be imaged in a reasonable amount of time. At that time, you might actually want to choose to image fewer genes. Now, you know, using our own experience of looking into the brain, very often that, especially with the help of the single-cell RNA-seq data, you know, selecting several hundred genes to image, you very often find that at least there you can identify the number of cell types de novo based on this several hundred genes, and then match that with the single-cell RNA-seq data, and then found pretty reasonable match, and you don't really find missing cells. And I mean, actually, very often we do find there are two ways. You know, you have sometimes the merfish will split the single-cell RNA-seq clusters, and sometimes single-cell RNA-seq will split merfish clusters. But the level of depth and granularity that you get with several hundred genes are often comparable already to this whole transcriptome single-cell RNA-seq data. And there may be a good question is, what is the number? You know, are there computational methods that help us to determine what is the number of genes that needs to be imaged? And there I want to emphasize again what Jeff said is it's not as trivial as one might think, because you might say, oh, then I select a subset of the RNA-seq data, and then I see how does it recapitulate the original RNA-seq data. There you find that, you know, it doesn't work exactly in the same way as when you translate that into an actual merfish measurement, because the signal to noise properties are different, and then the merfish data can be more sensitive. So by working into the different signal to noise properties and the sensitivity, and then determine what is a good number of genes to measure, and then ultimately that might be helpful for a large atlas effort, because you might not want to spend too long on imaging a single field of view.
That was great. Thank you very, very much. Does anybody else want to contribute to this question?
I'm just wondering if people have used prior knowledge about gene function in this problem, pathway information, and information about cellular localization of proteins and transcripts potentially as well.
Yeah, prior biological knowledge is often a very good thing to actually use. And especially for, you know, for people who are interested in particular functions and particular pathways. And those genes should be selected no matter what. And you don't need to actually just say whether they're good cell cluster marker genes or not, because sometimes they are very helpful actually for cell type identification, meaning that not only see that cell cluster, but also see what is the functional property of that cluster. So I think it is a very important thing actually to include prior biological knowledge, pathway information, and all those things.
Can you comment on how do you see the functional properties, or like how? Yeah, like how do you see the functional property, or how do you in practice incorporate prior info?
You know, there I think depends on different kind of biological problem you're going after. But if you use brain as an example, and then if you include some of the functionally known molecules, such as receptors, such as, neurotransmitter receptors, such as ion channels, such as the neuropeptides and hormones and neuropeptide and hormone receptors. Those ones you include that are very illuminating in terms of the cell type you identified, what potential function and how do they potentially communicate with other neurons. So these are, you know, very, very useful things to include.
I see. So I guess also one question would be, are we aware of like, so how interesting is to know the anatomical region where a certain cell type is in it. So anatomical regions, the boundaries of anatomical regions are defined with stain images and also with molecular markers like genes. And people are so there is an ongoing deep debate, and there is another session where people try to understand what is the right ontology, for instance, of the mouse brain or the other organs too. So one way to help is to be able to map and to find cell types using spatial transcriptomics, and then to register these images on existing atlases so that we can say that cell type X belongs to anatomical region Y. Do you think this is an important problem? Or like is anybody like working on this problem wants to comment on this particular problem? Oh, okay. And somebody just chime in, I think it's our facilitator, who says, back to the plan. Okay, says to take a break in 2 minutes from now. Yeah. So maybe.
Just to remember that you come back exactly to this Zoom room. You do not go to plenary, you come to this subtopic room.
Okay.
After the break. Cool?
Oh, okay. So we have 2 minutes more for some final comment.
I will make a comment on the probe on the gene expression probe selection. I'll make 2 comments. The first one, which might have been captured, I keep losing my Wi-Fi, so I miss some of the things people say. The first one is that I think in the single cell world, we have an increasing realization of the importance of, well, maybe re-increasing. It was increased, then people turned to look more at cell subsets, and now they're coming back to gene programs and the covariation patterns between genes. And that's a different way of thinking about genes to select for probes for in situ assays than by looking for genes that discretely characterize different subsets of cells. And that can work really well for situations like gradients, but also just for programs that are actually shared between multiple subsets of cells, and they'll never be chosen as markers for that reason. But they're very important to understand responses of tissue. And the second point that I would try to make is in favor of randomness, that you know, choosing random probes might not be a very bad idea, especially as the numbers of probes increases. And there's work, for example, from Brian Cleary, who used to be in my lab and is now an independent fellow, that shows, I think, that this is an interesting path to take, and it's especially liberating when you don't have single cell data, or when you are worried, as I think Joey and Jeff have said, about about percolating the limitation of one assay into another. So since we had 2 minutes, I think I just filled them in.
Yeah, and I think the scribes are like I can monitor them. They're like lively writing notes. I can see them here. So I will review all these notes from part 1, so that I will see you, hopefully all of you, for part 2, where we can start zooming in all the problems and questions that we raise here. Okay, thank you very much, everybody, for coming. Okay, so first of all, we need to find the scribes for part 2, which the simplest thing would be that the scribes of part 1 will also volunteer for part 2. What do you guys think?
I'm happy to help again. I hope my scribing in the first part was okay.
Yeah, was.
Thanks to everyone else who helped write notes too. So.
Alex, would you like to pass the torch, or do you think you can do the scribe for part 2 as well?
No, I'll continue.
Okay, great, thank you. Okay, so I reviewed the notes of the scribes, and so there are a lot of interesting things. So we started from like the importance of analyzing structured tissues, and then we talk about how to call cell types from spots and how to leverage non-spatial data to increase the accuracy. We talk about the importance of having a curated list of marker genes for all the cell types, and also how important it is to choose the right genes when they're limited in situ data. So when I ask everybody, like, do you think that the problem is solved? Do you think we can, call the cell types in situ data, or align them with single cell data. A lot of people said, well, this is not a solved problem, but we are working on it, or I know that there are several labs working on it. So one option for part 2 would be to try to address the technical challenges and to try to see how we can solve them. And that would be like one thing that I think is interesting. Another option would be to try to picture, to try to imagine that we solve these problems. So try to imagine that we are able to merge spatial transcriptomic data, and we have single cell resolution and a throughput which is 30,000 genes. At that point, we can start thinking of what is the most meaningful way to use this data. And so we talk about like how do we infer gene programs? How do we understand the disease? And somebody was chiming in with the occurrence of like occurrence of cell types. So how do we use that? So I would like everybody to maybe vote and say whether you would like to have a technical discussion on cell type calling or integration of spatial transcriptomic data, or whether you would like to talk about collaborating for the most meaningful way to use this data, assuming that technical problems are solved. Okay, so I'll start with Alex. Alex, what do you think?
I think I would vote for the technical challenge.
Okay, for the technical challenge. Viv, what do you think? Maybe we can move to Candace. Candace, lei. I'm not sure how I pronounce it.
Sorry. Can you hear me?
Yes, we can hear you now.
Yes. I would vote for the second one. Assuming we solve the first layer of technical challenges, what do we actually want it for? And that's for two reasons. First of all, it's a good motivator. We shouldn't be worried about the technical challenges if we don't know what we want to do later. But also often different technical challenges are more or less important depending on what you need the data for later. Such as which queries would you want to do on the process data. So that's my vote.
My vote also goes for the second part, but I like to hear from everybody. So Candace lei, and I'm sorry if I don't pronounce your name in a correct way. What do you think? Maybe we can move to Hiroyuki Aburatani. You're all mute, so I don't know if you're speaking, but if you want to contribute, you need to unmute yourself and say something, and then you can maybe. Okay, I'll move to the next one. Gellert Gau Blomme. Junil.
Hi.
Hi.
Just unmuted. I think this, in part one, I resonated with 2 points. One is what Peter Kachenko said. He felt the question of identifying cell types in spatial data is an empirical problem because cell types are hierarchical. You have a kind of a hierarchy of cell identity. The easier ones can be solved with even sparse data. When you go to middle of the tree or the leaflets of the tree, it will be increasingly harder. So I think that is a good answer. But one thing I felt we haven't followed up more is what Dana talked about, the interest to study developmental time course with either morphogenesis or organogenesis. That's really interesting because well, ideally we want to have just a continuous imaging for a week or for longer. But short of that, we need to have some way to align different images, ideally with lineage tracing, so that we at least know who is descendant of who. So lineage tracing lays down to the spatial data is something I would be interested in. I don't know whether it's off topic or not.
No, I think that's nice. I would ask you one question. So when you want to add the study of developmental time course, would you want to talk about technical challenges for doing that, or do you prefer to assume that we can do that and then we talk about what's the best way of using this information? And then I would count your vote in part 1 and part 2.
I think it's not a solved problem. It's even at the beginning of solving. I heard exciting talks on having a live zebrafish under the microscope and then doing real behavior tests and imaging the brain. So that'll be one rare example where you can do some kind of change, or even before or after addiction, if you keep the fish there for several days. But for most systems, we don't get to do continuous imaging. So be able to do this cross-sectional linked to a pseudo time course, but still know where we are and which lineage we are, seems to be a challenge, but really worthy of some consideration.
Yeah, no, thanks. So I was not saying that the problem was solved. I was interested in, like, would you like to talk about the technical challenges or how to use this information? But I would, I think both are interesting, right?
Yeah.
Okay. So Gary Bader, would you prefer to have the discussion on like technical challenges, or would you prefer to assuming that we can solve technical challenges, and how do we use complete spatial transcriptomic data to address useful biological questions?
Personally, I'd be, sorry, I missed the beginning. Personally, I'd be interested in delving deeper on a particular topic. If it's not, if people know about it, then to learn more about what's being done, or if nothing's being done in the field, to brainstorm about ways of approaching or solving a challenge.
Yeah.
So that would argue for the technical things, I guess.
Okay, Matthew, what do you think?
I guess my vote would be for the second one, the sort of, yeah, using that data to sort of how can we sort of solve the biological questions.
Okay,
Tommaso?
Yes.
Yeah, sorry, I was muted earlier. I will also vote for the second option. Okay. And perhaps the suggestion is that everybody can vote simultaneously. If in the participant window, you would ask people to say yes or no. So if you go to the participant window, there's a yes or no button. So if you ask a question, everybody can simultaneously vote.
Oh, this is very interesting. Okay, so okay, yes is part 1 and no is part 2. So it's not really yes or no, but so yes means I want to talk about technical challenges. So we will like dive into methods and algorithm, and no means I want to assume that I have complete spatial transcriptomic data and I want to understand what's the most meaningful way to use the data. Okay, you can vote now. And thank you so much for suggesting me. This is
actually, I actually, personally, I don't have care between the two. I could do one or the other, but I do agree with Gary that unless we do narrow either one or the other, because both topics are really wide and they're too wide. So I really don't care which one we do, as long as we set a narrow set of goals where we can actually make progress. And given this narrow set of goals, I would go with either.
Okay, from the vote, it seems there is a part 2. So so that would be, we do have tissues and we do have complete transcriptome information at the level of single cell. At this point, we should think of like, what are the most meaningful quantities that we can measure? And like some people were talking about occurrence occurrence of cell types, and some other people were saying to include like information about pathways to be able to extract gene programs. And somebody else, I think Dana, was talking about interaction between cells. So assuming, so the general question, and then we start diving in, but I guess an interesting general question is, assuming we have complete information of transcriptome at the level of single cell, most meaningful way to address 3 or like more subtopics. So one, I guess, since nobody's like really chiming in, so one thing would be assume that you have a tissue from, so now there is like, for instance, there's COVID, and there is like the lung map is doing like a big effort, and other consortia or part of the consortia too. But let's say that we have lung tissue from healthy people and sick people, and let's say that we have complete information of the transcriptome at the level of single cell. What would be the first thing that you would do?
So does anyone have any challenges that they've kind of already faced in this area?
I think one of the problems is that we are still very much dealing with the first half, the technical challenges of just dealing with the fact that this data has issues, and even just processing and figuring out what's the expression in different regions / cells is hard. And also the other problem is that this is a very specific thing. So, you need this is very biological. So biological is hard to talk in the abstract, which means like someone needs to really define, here's the data set, here's what we collected, and here's the question. And then we can start playing.
Can I ask this maybe in a somewhat different way? I think not just us on this session and in multiple other sessions, there is this strong sense that we absolutely need to have spatial data, or we're missing something critical. Maybe a different way of asking Tommaso's question is, what is that thing that one wants to learn from the spatial data? Surely it's not just to categorize cells by their gene expression patterns, because you don't need that for it. There's something else that people are looking for. What is that thing? Because, and the reason that I think that's important to ask early on rather than after you solve the technical problem is that for some of these questions, maybe some of the technical problems are not necessarily relevant. For example, if you want to know that the subset of cells tends to aggregate together, maybe cell segmentation is not the most important problem to solve. You can do it so, and you will still be fine for your problem.
Oh yeah, cell segmentation is overrated for most of the questions you'd want to answer.
I just gave it as an example because it is not overrated if what you wanted was to cluster individual cells. So I think it does depend on what questions somebody wants to answer downstream. That's why I personally voted for 2. I was very curious about what problems people actually want the spatial data for.
I mean, without going into a specific biological question, one thing is, I've seen all sorts of people do work with neighborhoods and saying, okay, this is the neighborhood of a cell, just counting the cell types around it. And I've seen people do very, very large scale, very, very clear annotations, such as, this is tumor and non-tumor, or this is like hot or cold tumor. And I've seen people say, okay, this is a gradient of this, that, or the other. But I haven't seen much method / language to really talk about spatial patterns beyond the most simple or spatial organizations among cells that are not this exact thing, not these are my immediate neighbors, but something more consistent, because the data clearly has a lot of structure, and I haven't found good ways to describe it. I've read quite a few papers, and some of them give something simplistic, but none of that really captures the spatial organization of the data.
I can volunteer to, or maybe one area of experience from the liver project. So liver has a number of cell types that are functionally divergent across a gradient, and the gradients are clearly anatomically defined. And from single cell transcriptomics data, we identify multiple subtypes of these cells, like hepatocytes. There are multiple subtypes identified, and we can use different methods to organize them along a gradient, but we don't always know a lot about the function. So we can obviously examine that looking at pathways from gene expression data, but if you look at the spatial information and even non-transcriptomics, non-spatial transcriptomics, but just any kind of spatial information, you start seeing morphological differences. You might see, obviously, the position of the cells along gradients is clear from that. But there are also, this reaches into another dimension, which is the dynamic aspect. So there's, for instance, liver sinusoidal endothelial cells have these fenestrations. They're little windows. It's pretty interesting. They're basically holes in the cells. There's lots of little holes, and those holes are regulated in different conditions, so they can open and close to basically let material pass through them. So they're kind of like a gate of some kind. So that is an interesting, challenge to kind of figure out what those structures do and how they work, how they change in different conditions. So there's like gradients and spatial structures that are interesting to map out and map to transcriptomics information.
And this is interesting also. Yes, please.
I was just going to raise the question, kind of following up from some of the discussions about, the vocabulary, the language of how we kind of like represent this data, and there's sort of like repeating units of kind of like individual units within a tissue, but there are also the kind of like non-repeating units which are at another level. So you can kind of like use the data to describe spatial information at different levels, and they're still at the microscopic level rather than the macroscopic level. So the kind of like the hepatic lobules, for example, which are kind of like repeating, and then there's the kind of like beyond that, which is how the whole kind of like liver microarchitecture is organized.
So to continue on this point from Maz, what I find interesting is that in biology, generally, structure, and especially repeated or conserved, or depending on time scales and length scales here, structure is usually a great clue to function. So the fact that you see something structurally repeating, in this case, it can be a particular arrangement of particular kinds of cells with their molecules in a particular histological structure, a different length scale. Things like that repeat themselves are often the first clue to function. And I think actually in the previous half of this session, either Jeff or Dana, or both Jeff and Dana, talked about if you find what the cells are making to each other, it's ways to perturb the tissue or to fix the tissue if it's a disease. So I think finding those repeated units and in disease, things that actually don't look like the repeated units, the destruction of them, is a great way for us for understanding function or getting the first clue that you can then follow up with. And at a level, not just of individual cells, but of multiple cells that work together to do tissue homeostasis. That, I think, the liver example is one example of that. The units of the gut will be the same thing, and so on. So even if you didn't know that there was a stem cell niche and a cell, an intestinal stem cell in the crypt, and it's sitting right there, the first clue that you would get for that would be that you just see that thing repeating again and again and again and again, always in the same position, always the same kinds of cells, always or roughly always in the same kind of organization. So you would say, aha, that's a unit to look at. What is it doing? And that's, by the way, what, anatomists and histologists and cell biologists have been doing for many, many years. We just have higher resolution.
Yeah.
There's another way of thinking about that by analogy to morphology of organisms, which was initially used to organize species, and then eventually genomics helped the molecular view of that. But it's not just the repeating structures, but it's just general similarities like number of legs or something like that. So I guess that's another way of thinking about it. I wonder if there's any utility of that analogy for taking concepts from that area of organizing species into this area. I'm not sure.
I guess I have two questions. So one question is back to what Dana was saying, like how do we capture the spatial patterns? So like if there is a gradient, like what Gary was saying, quite simple. I can see it at the naked eye. If I like just plot where the cell types are in space. But do we have a principal way to capture the degree of spatial organization of cell type? And that's question one. And question two is, how do we close the link from structure to function, which means like what is the best way, once that I have identified repeated units or gradients into cell types, what is the best way to make the connection between the structure of cell types and function?
So, for number one, I don't know, and I'd love to be corrected in this group, of any method that would be able to discover the crypt in a robust and reliable manner when you didn't know that that's the structure, when you didn't know that you were looking for a crypt. Of course, I can build an algorithm that will find the crypt, but like a priority, you don't know the structure and you want to look for a structure like, and find the crypt. I don't know of an algorithm today that can do that. I do think, and maybe that's because I'm downplaying, the power of computation, is the next step after we find these structures is I wouldn't try and plug it into an automatic algorithm. That's where we bring in the domain expert. We say, look, we found these structures. These are the cells. These are their expression patterns. These are how they change in different parts of this structure. And I actually think that this is where, we don't want to take the domain experts out of the game, but if we didn't know about the crypt and we could find and characterize the crypt and bring it to experts in that domain, then that could have been, an amazing finding. But I don't know of any method that can even find the crypt if they didn't know that they're exactly looking for that crypt.
So what about getting more histopathologists involved? Because there's a huge wealth of knowledge from kind of like centuries of H&E analysis.
I also think that the field of pattern formation, the mathematical field like Turing patterns, Fourier transform, Fourier-like transforms, they are capable of revealing order when it is not clear at the naked eye. I mean, it's quite simple for us to like plot the cell types and recognize that that particular cortical neuron stays on top and the other stays at the bottom, or that there's a gradient there. But there are like computational methods, a lot of computational methods in the field of analysis of patterns, which I think we can really use to answer Dana's question.
So
some of our experiences, basically, we took the approach of the hidden Markov fields and agreed to basically find the non-random patterns. We didn't have enough data, I think, especially like nothing nice with repeated structures like the crypt or anything like this, but we had all this available brain data. And I mean, the results were somewhat discouraging. We were also hoping to discover all kinds of, small, succinct structures. And it seemed like most of the things that were non-random that were picked up were basically larger regions or regional features that are pretty obvious, you know, so layers and so forth, that a lot of times were basically driven by the field of view limitations, right? So to get sort of a statistical sense for the pattern, you actually need to see it many times. And then, you know, if you look at most of the examples, at least like the brain examples, you know, you would have like one snapshot of that region, and you don't see it many times, and the regionality is pretty coarse. So while I think this is the right direction to go, it's not straightforward, and I think a universal algorithm like this will require a lot more data to start discovering things.
Yeah, definitely data. But I do think, I mean, again, I think we have a lot of our strongest methods, as you say, require quite a bit of data. But I, again, not with a clear idea, think that we can do much better than we're doing right now with the right idea. I mean, in the early days of gene expression, we actually managed to squeeze a whole heck of a lot out of that data. And the way we did it was not, you know, take a hammer from machine learning as is and just, you know, smash it to the data, because then truly there's not enough statistical power. And really, when I apply some of the methods that I've been trying to apply, they're really good at finding artifacts more than biology. But really, one needs to put into a method like that some type of biological insight, which I have yet to figure out. But it is very clear to me that any method that we devise that will work in the current data amounts requires some type of biological aha modeled into the method itself.
So Dana, I agree, but just based on what you said before, I just typed in a search, multi-scale unsupervised structure learning, and there's a bunch of papers that come up. I'm not familiar with the area, but people are using these types of things in satellite images, for instance, to identify multi-scale spatial features.
Again, as Peter said, most of the methods that were devised for other domains have a shit ton of data and just, you know, can't work. Again, Peter is right. We have a sparsity of data, and most of the other domains are swimming in it.
I mean, frequently those methods are still useful, but then can be modified to include ways of including additional data, right? So they provide good starting points to the approach. But also, I wonder if there's a way of collecting the data in a way that helps these methods. Like Peter mentioned, you have these small spatial fields, but maybe you have multi-scale imaging technologies that can do big spatial fields and then zoom in a kind of pyramid way that people typically think about these images in, and maybe that can be used as input into these methods.
Do you find adding information from histopathology helpful?
That sounds like it would be a good layer for one of these, you know, if you had a multi-scale learning system.
I use histological images when I like combine to in-situ data just to like to see where the cell types are. Like I use them to orienteer myself.
The other thing is that often you simply have a lot more histological data that pre-exists. So if you're in a domain like a disease domain where there's more variation between samples, then you're not necessarily limited to the smaller number of samples that you can do with the sophisticated experimental method, but you could in principle go to a much larger set of samples that show you a lot more variation in them in the histopathology sections. But you would need access to them, which is often not as trivial as people might think.
This is Andrew. I don't barge in. I'm just, might you miss something from that? If you're looking at that layer, it may not show you the structure that you see in some other modality, right? You might not see that underlying. It may help in some situations, but you may also miss some underlying structure. Anyone following what I'm getting at? Right, if you just look at the histopathology, it's not going to tell you anything about necessarily the gene expression. You may actually miss something in the structure there that you're looking for.
I think the comment was directed at sort of multi-model imaging in a way, right? If you can do, let's say, a MERFISH or something like this at very high resolution, but also for the same region have, let's say, histopathology, you can start to use like traditional histology stains to kind of connect things. And then maybe, you know, learn more from histology. But at some point, you need to basically bridge it with high resolution.
Back to your comment. Yes, please. Junil.
Yeah, I have a comment. I really appreciate this discussion, I think, because it's very conceptual. We talked about, we actually don't know where is the biological variation being driven, which layer it is at, so that's multimodal. We also don't know which scale it is, because sometimes pathology could be subcellular, or sometimes it's a cellular. Other times it's like, you know, 5 cell or 50 cell scale. So we are grappling with the language to talk about structure, or the norm of the structure and the variation around that norm. Only then can we talk about the pathological departure from that norm. And then this kind of needs to be built on, indeed, all spatial scales, not just one cell, but also there are 5 neighbors, there are 50 neighbors, and then 2 different directions. You know, the space constants are different. So I think Dana's framing of the problem are, how do we extract some quantifiable features on all scales? I remember my colleagues who study just regular image on wound healing, they say, you know, after you look at so many of this, you have a gut feeling, but it's not formalized. But if we press ourselves to be formalized, we would have to be talking about coloration or the edge roughness or some kind of spatial entropy. So those are the attempts to develop metrics to talk about structure, you know, what's normal and what's a departure from it.
And actually, I just want to chime in and say what June just said. So Peter, you were saying we do need a lot of data to be able to train this method, but there are, in the field of mathematical analysis of pattern, which is different machine learning, we do not need so much the data, and there is a lot of insight that we can get. Just the method that Junli said, like spatial entropy, is a very smart way to look at patterns, and it works even though we do not have a large data set.
I think my comment was in terms of what we were able to discover with this, right? Because I mean, I don't think it was a shortage of the algorithm. There are plenty, and we should certainly look into them and keep an open mind about it. But realistically, if you approach the, let's say, analysis of the cortical region using this type of, you know, these spatial learning methods, you're basically going to get the layers out, right? And all this sort of specific kind of predicted interaction specificity is kind of going to reduce to the layer structure, at least in our example. So my comment was that from the amount of data and the types of examples that were at least, you know, publicly available, we couldn't find something to dig into. And I think with time, and now as more and more labs are doing these measurements, there will be more interesting collections to dig into, and then we can actually see how these methods perform.
I guess one thing that might be nice for the machine learning is to have available data sets that are fairly clean, that many people can work on, and that could be a specific action item. Frequently in that space, there are nice competitions that people develop too. I don't know if people have data that they're sharing already in this space.
But to follow up on that earlier comment, I do feel, you it's not the time to feed all the data to a black box model to shake it out. We want to learn a lot of the mid-scale features. You know, those who look at histological graphs now try to do classification, but also try to have interpretable features for those classifiers. For us, I guess we are at the threshold of learning both single cell, morphology, projection, size, but also their neighbors. There needs to be summary statistics about the neighborhood composition, I guess, because I've seen algorithms presupposing cells that are alike aggregate together. I think the opposite could easily be true, that cells that are not alike may prefer each other. So these are empirical questions, but we still need metrics to score an image or the subdomain of an image for these kind of emerging features.
So I think, so we name the metrics that we use. So we all think it's important we come up with good ones. And we all think that we need more data in order to be able to use these metrics and like extracting meaningful patterns. Shall we try to dive into like some details and like try to metrics that we use? And I can start. So once we find type, so one question is the granularity of types, which means do they tend to concentrate all of them in one area, like the cortical layers that Peter was saying? Or do they tend to like distribute in space? And I'm thinking of the microglia. And so like, okay, so I guess one metric for me would be the level of clustering. Once I have the probability distribution of a certain cell type, and assuming I have enough throughput, enough samples, so that my statistics make sense, one quantity that I would compute is the level of clustering, which is informative for me for that particular cell type. Does anybody want to comment?
Can you define level of clustering for me? I don't quite understand what you mean by that.
Yeah, so the specific details of how there are several ways. I can tell you two, which by no means are like the right ways, but this is what I do. So one way is to compute the Shannon entropy of the distribution. That would be the integral of p log p, and this quantity is very, very, very small if the distribution is peaked, and is very, very large if the distribution is broad. So
this is distribution over the cell types or over the cell types in a given region, or?
This is the distribution of one cell type in space. So if I have the motor cortex and if I have like a cortical layer, I have my cell completely concentrated on the top, then the Shannon entropy is gonna be very large. Otherwise, it's like the microglia, which are distributed, it's very, very small. I would just I would just tell you about the second method. The second method is machine learning based, and we know that there are algorithms like the means that find cluster. So one is to run this algorithm and see how many clusters they find. The number of clusters learned is one way of learning the clustering of a cell type. Yeah, just some thoughts, but please chime in.
We use some normalized Ripley's K function for this type of thing, which kind of like a point process spreading, but just another measure. You could come up with several.
Sorry, can you repeat that?
It's called Ripley's K function. It's like a.
I never heard of it.
But yeah, I agree with you in general. This seems like a very basic characteristic. Do you find these cells clumped, or
yeah,
do you find them distributed?
Yeah. And also, this is one metric, but there are others. Like clumpedness is only quantity we are interested in. So I also expect people to chime in and say, oh, but we are also interested in this other thing, and there is this metric for this other thing.
Well, layering is the other one.
Yeah.
I don't have a good metric for this. We had this in the hidden Markov fields. We had sort of the inertia term that we could analyze, which kind of basically picked up projected patterns, like layers, directed patterns. But it's not very well worked out. You can probably do something simpler than this.
Does anybody else want to propose? So one other metric that maybe people can comment on is to investigate the occurrence of cell types. Like, I'm not only interested in if that cell type is uniformly distributed, but I also want to know whether this cell type likes to stay near to some other cell type. So what kind of, do you think are the right way to understand that?
Well, here you can use, covariation is a very wonderful metric for that type of thing. And another thing which, I'm really surprised that we don't do more, and again, guilty as charged. Only recently have I started to really dig in very deeply, because up until now we were, as I said, in the weeds of the more technical things. But, we have imaging information. And so that allows us to sort of figure out what cells are next to what cells and sort of build a graph neighborhood or whatever. But I've never heard, and again, I haven't read all the literature, the morphology of the cells. And I've actually seen, the morphology of cell types that, at least when we look in with a few markers, so maybe it's a different cell type if we would have had more markers, but the actual morphology of the cells. And we see that particularly in our MIBI images, where we get morphology, which I guess you can't really get in transcriptomics as well. You really see changes in morphology. I don't know if you could even get morphology from sort of transcriptional data, but certainly, do at least some protein cell membrane with transcriptional data. Cell morphology is a huge cue.
No, I didn't. I think I want to say something.
Can I bring the question again about histopathology? I think I raised this a bit earlier about pathologists having like a well elaborate nomenclature and everything. I mean, our epithelium is sort of like described by morphology, like cuboidal, columnar, and so on and so forth. Why are we not, taking advantage of all of this information as kind of cues or incorporating them in the analysis?
I think right now, most of the collections haven't been coordinated enough, so you get, the histology along with, again, we are collecting histology with every image, at least in our cancer project. So, I haven't seen a lot of that data published out there, where people are doing both histology and the more multimodal transcriptomics methods. I think part of the problem, particularly with the transcriptomic methods, is pathology is this FFPE based world, which is very different than the type of tissue that you need for the transcriptomic world.
So I'm a pathologist, and I'm highly interested in the spatial transcriptomics area. So of course, we look at FFPE, but we also have, like, for example, at our institution, fresh tissue collection effort on frozen samples. And like, for example, what I study is cutaneous lymphoma, and one of the tumor I'm working on is transformed cutaneous lymphoma, which by definition, for pathologist definition, the cells has to be 4 times the size of a normal lymphocytes. So for me, having morphology is extremely important. Incorporating morphology into a spatial transcriptomics data is very important.
Maybe a recommendation that can be part of this group is that collection should include more pathology and
yes.
as a like a thing. I think we have some recommendation for this somewhere in the HCA white paper, but I don't know how many people actually bother following that.
We totally should. I think for single-cell RNA-seq studies, it's harder to incorporate that information, but for spatial, I think it's totally needed.
I agree, actually. I think this should be kind of like one of the, outcome points from here, and perhaps like getting detailed histopathology report on those sections is going to be really helpful. And from there, you can then learn, and subsequently you might find that you don't have to have such big data sets, and you can derive that information, and then you have bigger, and you've learned from this, and you can kind of like apply it in the future where you don't necessarily need so much histopathology input.
I would say three things. First of all, yes, Dana is right, it's in the white paper and it's actually really important. The second is that people often assume that if they wanted a histopathology later, what's the big deal? People do it all the time. Well, it turns out that if you have no specimen left and you didn't think about this in advance, you would not actually have this information later. You will have used the sample. So it's something that's really important to either do in advance or at least preserve the tissue in a way that allows you to do it later. And also from what we see already, just with bulk RNA-seq and histopathology, as well as with other contexts, it's probably going to be the case that as we learn better and better the relationship between something like an H&E stain in these higher, more sophisticated molecular assays, you might not need to do the sophisticated molecular assays every time. It might be that you would have a good enough model that would tell you, given something simpler like an H&E stain, a lot about the molecular content of the image. So there's also value downstream in building these good training data sets to learn models that are more and more sophisticated about how these different types of information map to each other.
And it's relevant for the cell-cell interactions as well, because even with H&E, histopathologists can actually identify like the different cell types without needing the markers.
Exactly, and that means that there's information there. We just need to learn the mapping, and also to Dana's earlier point of bringing in the experts. Many of the experts are well trained at seeing things in those images, and so we could use this to supervise algorithms, but also to just help with interpretation later on.
Yes, for example, in the tumor microenvironment, I want to see when, for example, a large 4 times large transformed cell is next to the normal lymphocyte, how do they interact? How do these cells communicate at, for example, the receptor ligand level?
Can we name one data set which we can access to that contains both histopathology and spatial transcriptomics? I mean, I can name a few, but maybe you have better ones.
So the way I have been collecting tissues from my own new study right now is I split the tissue into 3 pieces. 1 piece is for single-cell RNA-seq, 1 piece is net frozen in hope of pursuing spatial transcriptomics, and 1 piece is FFPE.
So I'm like speculating here, but what I think is that so somebody asked, maybe it was you, why don't we use histopathology and why don't we consider it in analyzing spatial transcriptomic data? And I guess that the answer for most computational people is we are currently limited by the data, and we lack a comprehensive data set where we have either one and the other so that we can start playing with this data. So if there are experimentalists here, and if you know of a data set that is accessible to the people in this workshop, you should stand up and speak. That would make the life easier for a lot of people.
I think this is what most experimentalists would plan moving forwards. I don't know about the existing data sets, but this is generally where the direction of travel will be for most people, and you'll find that these data sets will be generated. What will be quite important is how we then preserve the metadata or the data relating to the histopathology. that will be the challenge in terms of how you kind of like have that as the accompanying data set to the, you know, high dimensional analysis.
So I think we have a few minutes left. Let me just check. But essentially, I think where we are leaning toward, where I'm gonna. So now, what I'll just speak like for a couple of minutes, and then we will keep talking. But I think I have them to write something in a blue box, and I think I'm leaning toward the following story, the following picture. So we really want to understand function. We really want to leverage spatial patterns of cell type to understand function. We know that there are spatial patterns like gradients in liver cortical layers. And we agree that one, we need more data. Two, we need to be able to integrate data across different modes, like for instance, histology with spatial transcriptomics. And from the computations, we need to come up with metric and indicators that would be informative for us, both in detecting patterns that are not so visible, or in providing a quantitative assessment like of these patterns. So I guess one effort where this community can interact and collaborate is to develop such indicators to identify and describe spatial patterns in order to understand function. I think we have a few minutes left, like 10 minutes. So probably you want to, I don't know, comment more on that, or?
I actually think your summary is very good and extremely well done. I generally think that you moderated this very well, so kudos to you.
Thank you. This is the first time I'm moderating. I'm so scared. But,
well, you're doing a good job.
Yeah, Tommaso, you did a good job. I just, in the chat, posted a link to a paper that I heard a talk not long ago. It's about morphology. It's quite an ambitious way of classifying neurons using the mouse brain atlas data.
Yeah, so thank you. I will read this.
I guess one thing that I would raise before closing down, just a quick poll. I mean, for me, a lot of the things that are done in the neural world, because I don't know, they're very, because of the way the spatial organization and shape of neurons, it always feels to me that like the brain imaging is sort of another world. And while they're the most advanced for sure, their methods are often less relevant for, let's call it, more epithelial tissues and just more regular internal organs. Do people tend to agree with that statement?
I agree.
Yeah, I agree with you, Dana. I think different organs really have different, prototypes of shapes and local structures.
Well, there's different structures, but I can imagine a method that will work generally on the different internal organs. That, the structure will be different, but I can imagine a single method approach to treat the different internal organs, maybe with some organ-specific modification, but like some general similarity in the gist of it. Whereas for the brain, I feel it's like almost like it's an alien organ.
Yeah, I agree that there are probably a countable number of prototypes. Brain is highly laminal, and then area, then it's folded. Then there are other places that's very tubular, right? It's just a cylindrical coordinate. Others are apical to luminal, so that's a principal gradient, sometimes with crypt in it. And then the scale will be different. If you go to liver on the largest, you say this is the proper shape of a healthy liver. Then you go in, there is donation. Then you go in, there is a hexagonal structure. I guess that on each scale, there needs to be local statistics to describe what is the range of norm.
Yeah, I do think that's actually a really important point, which maybe you can incorporate. The, you know, what is the range of norm? When is a structure? I think one of the most important outputs of the sort of pattern structure organizational principle detector would be not just to arbitrarily find patterns, but to quantify these are the patterns within norm, and this is when you deviate from norm. That would be like a thing these patterns can do, and defining patterns and the range of norm within that pattern, I think, would be an incredibly wonderful thing to do.
And another prototype shape would be the vasculature or the arborization of airway in the lung. They are almost like a fractal. So that's a kind of a shape primitive.
So like speaking of patterns, like I'm also part of the HubMap consortium, and what we are doing there is that we try to generate airways looking structure using fractal geometry. And once you do that, you then learn that there are several exponents for these fractal patterns. And so learning that number is one other metric that you can use that works incredibly well to characterize a spatial problem. So that like one other metric that I was just thinking of. So we do have 10 minutes left, 10 minutes more. So I think we want to use all this time. Does anybody wants to add something, or I will throw in some questions?
I'd like to add a few more comments. I've recently been reading more about weather prediction, how that data science ecosystem has tried to integrate over multiple scales, as you can imagine, from the molecular collision, the physics of it, to the mid-range, what you call like temperature and pressure, and then to the larger ones, you know, cyclones, fronts, and they have to grapple with on which granular size you can compute, but also on the Earth scale. So on the largest, they call it Earth system data science. We don't really have an equivalent for like human body, whole physiological system data science, but they want to do this fairly comprehensively for mid-range weather forecasting. So say for the next Sandy, whether it will turn to New Jersey or not, they have to know, do they want to do a cubic of air 9 kilometers in size or 18 kilometers in size? And then how to lace in even finer data by, say, fly an air balloon in, so that they have, for the crucial points, to get higher dimensional data. So that's equivalent to us talking about having this orthogonal, some are, you know, space poor, time dense, others as opposite, allow more orthogonal imputation, I guess, to really learn the structure and its dynamics. So I guess my comment is there is a lot to learn from these other domains, and some are ahead of the biological field. Yeah.
So that's a great comment. And in the study of ecosystem, there are also quantities like a species area relationship, where people try to understand like the number of species in a certain area, rank abundance distributions, and sometimes they obey very simple laws. And there is the Hubble theory of neutral diversity, which is one of these famous theories that people have been like talking about. And so I also think that these particular indicators are very good metrics for our patterns, where we substitute the cell types with the species. I'm not sure. Did I understand correctly what you were saying, or did I?
I think so. It's really how the diverse field working on different scales. Like for us, that we have biochemists working on, say, RNA granule or the condensate in this or that part of the one cell. Others on single cell, others on developmental biology or physiology. But for people who do midterm weather forecasting, they have to decide when the mechanistic model, physics-based model stops, and then just large data pattern recognition starts. So the handoff between the unbiased to the knowledge-based model is something they have grappled with for decades. Then I think it's healthy now that they have improved the forecasting power even compared to 10 years ago. It's not a blind chase for larger data or just higher computing power, but it's kind of a strategically placed, balanced investment on different types of data.
Do we have more comments?
I think on that comment that was just made, I think for many biologists, what's interesting is how these two levels connect or couple. So it's not just about making the forecast or making the prediction. They actually want to understand how the molecules make the cell or the condensate and how the cells organize in the tissue. They don't want to let go of the underlying level of biology. So that's why I think it's a difficult thing for, it's almost like it's very difficult to sell that. It's sort of not the right explanation for many biologists. If you say, well, I don't care about what happens with the molecules, I'll just explain it at the higher level, they come back a little dissatisfied. That's been my experience with it.
Okay, thanks. This was nice. We do have a few minutes for some final comment before we will be disconnected.
I have one question, which is going back to the competition idea. So machine learning, a lot of machine learning research is driven by competitions over shared data sets. They have boards where all the scores are tallied, and it's a good way of engaging the machine learning community. I'm just wondering if that would be if people think it's a good idea to somehow engage a machine learning community by making data available in a particular way with a particular challenge, kind of like Kaggle or something.
So there are already.
I really think that at this point in time, we don't have enough data for these sort of big, heavy hammer-based methods. I really think we're still at a point with the amount of data that we have that I think the people that have domain expertise have a better chance at it. Of course, collaborating with someone who's hardcore machine learning is great, but they really, I don't think we have the amount of data to really just throw it at them. I mean, a lot of the machine learning approaches are very, let's call it, supervised, and we haven't figured out what we want to supervise yet. So I really do think that, working collaboratively with machine learning people is awesome, but just throwing it at them and telling them, here's a challenge, solve it for us, will unlikely work.
What I keep hearing, because I jumped around a few sessions earlier, is that we need more data, and it makes me really eager to get back to the bench from home. So everybody wash your hands, please.
So there are some Kaggle challenges, like for with biomedical application, but mostly they are for segmentation. Like they have CT scans of lung, and they want to segment fissures and lobes. And I totally see Dana point. Like we don't have enough data to crunch big neural networks yet. Okay, I think we have, in 5 minutes, we will be automatically kicked out. So I think I know what to write in the blue box. So this conversation has been fantastic.
Tommaso, Tommaso, I was just thinking about your last point that you said Dana is right, we don't have enough data. Should we come back and say what is enough data? Kind of as a challenge to the experimental community, saying that's the scale of something that could engage, say, the machine learning community more successfully for us. We definitely engage them with single cells by giving them a lot of them. And,
yes.
I think we also have to have clearer questions.
I'm joking a little bit.
Sorry, Dana. I think we also have to have clearer questions and not just the poor dump of data.
I fully agree, but I think that's something that we need to actually tell ourselves, or we won't get something in return.
So are you thinking of like, should I write something like that in our blue box, or are you thinking of like?
I was challenging this group to say whether they think it's a good idea.
Okay,
to try and set some bar or some target or some goal like this.
Okay, we have 4 minutes left, and each experimentalist should chime in.
Shall we do the vote, the yes or no vote? Because I completely agree, but I just think, we might be able to just do the vote and you can see the answers.
Okay, please lead and state what yes mean and no mean so that everybody can vote. Yeah. I think it would help if you repeat. No, people are already voting. Okay.
I assume is yes, we need to define how much data we need from the experimentalists and give him a target goal.
Please, Dana, go.
Looks an overwhelmingly green. Yes.
Although the voters are like 30%, maybe 40% of the participants. Maybe the people who voted no can chime in and say why no.
Might have been left over from the last vote. Some people are also just observers.
We have one no, that's it.
Kinma, would you like to unmute yourself and tell us in 30 seconds why you voted no?
Sorry, it's a mistake. Yeah, I didn't mean no. Yeah, sorry about that.
Don't worry. Okay, so okay. So we all think we should like come up to an answer to that.